Hey, AP Bio folks, just checking in with you here. Wanted to give you a quick little down and dirty look at anaerobic respiration and fermentation. So, if you would do me a favor and pop in and grab the kind of skeletal notes to follow along with this, I'll try to keep it under 10, 12 minutes, and then that'll be sort of our wrap up of all the stuff to deal with cellular respiration. We've already talked in full detail about the aerobic version, and you'll be doing a lab in class to clarify all that. But what I wanted to do is just touch on the anaerobic version, which is anaerobic respiration and fermentation. So you can see on this slide right here, what I'm showing you is the pathways that y'all need to know. Right now, we are quite good at looking at starting with glucose, breaking it down with glycolysis into the two pyruvates. Those pyruvates then go through that link reaction in the presence of oxygen. They go through the link reaction or pyruvate oxidation to create acetyl-CoA in the matrix of the mitochondria. That enters the citric acid cycle, goes through here, creates a whole bunch of NADH, a little bit of FADH2. Then it goes on to the electron transport chain and chemiosmosis to do the oxidative phosphorylation to produce all that ATP. We know that. But what happens if it is anaerobic? Well, it's going to go from pyruvate, that's kind of our juncture. And at that junction, what's going to happen is if there's oxygen, we know what happens. If there's not, then we're going to do fermentation. And depending on what you are, if you're a bacteria or like a yeast, you might produce ethanol. So you might do alcoholic fermentation. Or if you're humans and mammals like us, you are going to do lactic acid fermentation. So here are my two targets for y'all. One, you're going to be able to explain it and what happens when there is no oxygen. How do we produce ATP? You can go down this checklist later and see if it works for you. And then two, as a finishing thing, how we always talk glucose, but I do want to talk about briefly what happens if it's not glucose. What if you're digesting proteins or fats? How do you get the energy out of those? So that's kind of where we're at. So let's kick it off. A couple questions for you. First off, again, the whole reduction, oxidizing stuff. This plays a key part in why your body does fermentation. So my question to you is, what is the oxidating agent of glycolysis? And if you look in the diagram, right, here's glycolysis, here's all the aerobic stuff. What is happening is this is being turned into this. Remember, this is our empty piggy bank. This is our full piggy bank. Then this gets emptied and returned back to here. So it's very cyclic. So our answer for the oxidizing agent of glycolysis is NAD+. What is the oxidizing agent in the citric acid or the Krebs cycle? Well, we're producing a lot of it. It is still the same thing. We're saying NAD+. And what is the oxidizing agent of the link reaction? Which again, we're producing more NADH. It is also NAD+. So remember, NAD+ is accepting the electrons, right? It's accepting them. It's storing those energized electrons for use later. So we have to be able to empty them. All right. So that is our key. So my ask is, once we have all that, well, how do we get it? Right? How is NAD+ obtained to run all of these reactions? If we need it for each one of those, glycolysis, the link reaction, the Krebs cycle, how do we actually get it? And this is the actual answer right here. Right? We oxidize NADH and FADH2 via the electron transport chain to make NAD+. Right? It's that cyclic nature. So this will get kicked into here. This will get emptied back to make more of this. Again, cyclic. Okay? My question to you is, without oxygen, what happens to the regeneration of those molecules? If we don't have oxygen, so if we say no O2 equals no ETC equals no NADH to NAD+, if we don't have any NAD+, then we can't do aerobic. We can't fill more because we haven't emptied them. and we're stuck. We run out of ATP. So if we do that, what happens? So my question to you then is, what if we don't have enough oxygen? Without O2, mitochondrial reactions will shut down. Remember, I gave that kind of silly analogy. I said it's kind of like that conveyor belt at the Cub Foods or wherever you do your shopping, right? This will keep going and keep going and keep going. And if we don't have oxygen to make that water, this whole thing will back up and back up and back up to the point where these will not get emptied. So we won't get any more of that and we won't get any more of that. So that's what would happen. We know that without oxygen. So the question then is, if we stall out, if we run out of oxygen, if we quit emptying our NADH and FADH2 to make NAD plus and FAD, how can glycolysis continue? How come we don't die? Because we don't have enough ATP. And we know, hopefully you know, the answer is glycolysis can continue because our bodies can kick into fermentation. Now, we cannot maintain. That won't last forever. We need more energy to survive. But for short periods of time, until oxygen becomes available again, we can continue glycolysis. How does that work? Well, here you go. These two images are those exact ideas, all right? Basically, what you're seeing here, these are unlabeled images from your textbook, but what you're seeing, each one of these is a type of fermentation. This side over here, this is alcoholic fermentation, and this one is lactic acid fermentation, okay? We still start with glucose, just like we know. We start with glucose, just like we know. Something comes in. We have ADP plus those phosphates, ADP plus that phosphate to make a little bit of ATP. And again, we know it's a net gain of 2, right? So we know that. So if you look, got my ruler in there. If we look, this half and this half of the process are exactly the same. Alcoholic, because I can't spell, alcoholic fermentation and lactic acid are the same. We also know that right here, right, what do we have coming in? We have NAD plus and NAD plus, and we're making NADH and NADH. Now, here's where the difference comes in. This part here, taking glucose, putting in some oxygen or putting in some ADP to make a little bit of ATP, we get this and this. They're exactly the same. This is your pyruvate, right? As is this over here. So this is where they diverge, right? Between alcoholic and lactic acid. In alcoholic, we're going to pull off that highlighted CO2, and we're going to be left with this right here, right? This thing right here. There's a name for it. Don't have to worry about it. What we care about is that this will then empty. The NAD plus becomes empty. That H, if you look, where did it go? And again, simply look at the chemistry. See, there's the extra H. See, it's missing that H. So we take that H and we fill it in there. And what we make is an alcohol called ethanol. So lactic or alcoholic fermentation gives you ethanol and CO2, right? And again, if you know much about fermentation or brewing processes, right, that's what happens when they make beer. The CO2 bubbles off and gives it carbonation. The ethanol is the alcohol that's left in there making beer or wine. If you bake bread, you are doing alcoholic fermentation with those yeast, right? And what happens is the CO2 bubbles off and makes the bread rise. And then when you cook the beer or cook the bread, the ethanol evaporates out because the temp that it does that is much, when you heat it, that's what you're hitting. You're hitting that temperature, it evaporates off. So the bread doesn't taste like alcohol. So that split, right? This is us. what happens to us? Well, we still make those pyruvates, but now if you look, what happens is the pyruvate empties out the NADH, and again, you can see it's just a simple chemical on that bottom carbon with the double bond to the O. We add an H that came from NADH, and that empties NAD plus so that it can go back and continue to make 2 ATP, which is better than nothing, right? This is lactic acid, right? So it's a pretty, not simple, but it's a process that takes that and makes it happen. Now, I'm not going to dig into this. I just wanted to show you that there is a conversion back. You're not going to be responsible for knowing this. It's called the Cori cycle. But what you can see here is I drew a balanced pyruvic acid, which changes into lactic acid, right? We take those H's and we add them in here, right, to empty these. Well, what can happen then? Lactic acid, once oxygen becomes available, it's going to take that O2, it's going to split it and split it. It's going to take another NADH and it's going to pull out 2 waters, and it's going to convert lactic acid back into pyruvic acid. Right? So it's a crazy way for your body to store up that lactic acid and then convert it in the liver back into pyruvic or pyruvate so that you can then kick that off to the citric acid cycle and actually continue the process. So again, you won't have to know this, but it's called the Cori cycle. I just thought it was kind of cool. So there's a version of it to show you. Anyway, the important part is that you get the basics of why fermentation happens. The other part on here that I wanted to show you, and you can look in the chapter, I forget what it is, chapter 9.6. It talks about glycolysis and the Krebs cycle and all of that, and how we don't always. So we've studied this right down the middle. We started with carbs, we broke them into glycolysis, and then we had these different things in the middle to create pyruvate. That pyruvate made acetyl-CoA, that went into here, that made this, right? So that's kind of the one that's always studied, right down the middle. But what if your diet is low in carbs, but you're high in fats? Well, fats are kind of crazy because fats break down into, right, 2 different things, glycerol and fatty acids. Glycerol, there's an intermediate in glycolysis called glyceraldehyde 3-phosphate. Again, not important, but glycerol can be converted into that, and then it can enter the rest of the cycle. The fatty acids, they're long carbon chains, right? They're broken down into these two carbon compounds, which then join up with this to make acetyl-CoA, and which can then enter here. So you can still get ATP as an end product, but you enter at a slightly different place, right? Same can be said on the other side here for proteins. Proteins, if you have a high diet of protein, not a lot of carbs or fats, you get an excess of these amino acids. You break them down in your digestion, and you have these amino acids. Well, amino acids are a bunch of carbons, a bunch of oxygens, a bunch of hydrogens, but there is some nitrogen. So they have to, before they can do this, there's something called beta oxidation, and it essentially breaks down those fatty acids. Sorry, that's over here. That's beta oxidation is what it's called. But over here, what you have is a process where it's called deamination, right? Deamination. And again, the power of words, we're de-aminoing the amino acids. So we're removing that NH2 group as NH3, which you might remember is ammonia, and this leaves as urea. Once you've removed the nitrogen group, then the rest of it can go into different aspects. It can go into the end of glycolysis, it could go into the link reaction, or it can go into the citric acid cycle. So it's just a way of showing that it isn't one metabolic pathway. There's actually multiple ways. Carbs are the easiest to understand. That's what we looked at. But you could use fats and use their individual components, or you could use amino acids from the proteins, remove the amine group, and then kick them off into here to produce, because the end product is we need ATP. Right? So it's kind of cool to see just that example. And then finally, the last slide on here, last couple slides. This is showing you sort of how do we have a metabolic, what is our metabolic control? and they make it sound really technical, but it's really simple. It's called a feedback inhibition. So, what happens, and this is really simple, in a feedback inhibition, the end product of the anabolic reaction, which is ATP, inhibits the enzymes that control the breaking down of glucose. In other words, if you have a ton of ATP, your body's not going to waste energy breaking down more to make more if you're not using what you already have. So the production of ATP and having excess will inhibit an enzyme called phosphofructokinase, which is one of the enzymes in the 10 steps of glycolysis. Again, don't go crazy here, but it'll inhibit that, which will stop it from breaking down to make pyruvates, which will slow down the process. Right? There's also another one that if you have a lot of citrate, which is the entrance, that's the 6-carbon compound in the citric acid cycle, that will also shut off the enzymes from breaking up more glucose because you don't need to. So it's really pretty simple. This enzyme right here, they call it the pacemaker of cellular respiration. It is what controls the pace of the process. Right? It's an allosteric, if you remember that term from our earlier stuff when we talked metabolism, it's an allosteric enzyme. As it builds up, it'll block in a different site, it'll block it from doing more of that process. So again, it's just a simple feedback inhibition. They make it sound fancy, but all you got to remember is too much ATP is being produced, you're going to slow it down and you're going to stop the process until there isn't enough, and then it'll allow it to continue. Right? Not too crazy. All right. Wanted to keep it relatively short. Went a little longer than I wanted to, but hopefully that helps you understand. If you have questions while we're working on the lab here over the next few days, we certainly will have time to chat. So hopefully you have a good evening, a good week. And remember, test is coming. Thanks. Have a good one.